It's live.
Yes. Welcome everyone. Welcome to Bioingene webinar series. It is our 27th webinar in this series, and we're very happy that today we have Dr. Aryadeep Choudhury as a speaker. He's working on rice for more than 2 decades, and he is a very good writer and editor of many books. I request my friend Liberty to introduce him and a little bit about our bioengine, and then Shoma will interview our speaker. I request all our viewers. I already sent the feedback link to your email address. If you not get it, you can request by mail with today's password. This link is inactive for a security reason. We will start the feedback link, and before the distributing the password for today's webinar feedback and certificate application. This certificate will be available on 2nd of January from our website, bioingene.com. I request Liberty.
Thank you, Shubha. Welcome to the Bioingene webinar series, everyone. This series serves as a platform where plant scientists can present their research to the world, while future scientists can gain knowledge, perspective, and inspiration. The appreciations and suggestions that we receive from our global viewers keeps us motivated, which has led to the constant growth of Bioingene with new viewers and new endeavors. One new endeavor we are very excited to announce is the Bioingene Plant Sciences Journal. This journal is an online, international, open access, peer-reviewed scientific journal that is published monthly. We seek to publish papers that are relevant in the field of plant sciences research and address the present and near future scenarios of crop health, yield, and production in different biotic and abiotic challenges. Aspects such as food and nutrition security, effects of climate change, crop resilience, applications of AI in agriculture, genome sequencing, genome editing, omics technologies, CRISPR-Cas, as well as modern breeding techniques in plant sciences will be highlighted in this journal. Selection of manuscripts for publication will be based on novel research, original data synthesis, or concepts without plagiarism and violation of any copyright. Please visit our Bioingene website for more information. Now, back to today's event. This is our 27th webinar of the series and the last for the year of 2020. We welcome all the viewers who have joined us from around the world. After attending today's talk, you can apply for a certificate of participation via the feedback link and password. We have already emailed the feedback certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the Q&A and interview session after the presentation has finished. Submission of the feedback form is mandatory to receive a certificate. When you fill out the feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through email. Please visit our website to register for upcoming webinars. You can connect with Bioingene on Facebook, YouTube, Twitter, and LinkedIn. Now on to today's lecture. The topic for today's webinar is Journey of Rice Research from Salinity to Fluoride Stress Responses by Dr. Aryadeep Choudhury. Dr. Choudhury is presently working as assistant professor at the Department of Biotechnology at St. Xavier's College in Kolkata. He completed his Bachelor's of Science with honors in botany from the Presidency College of Kolkata and a Master's of Science in Biophysics and Molecular Biology from the University of Kolkata. Thereafter, he joined Bose Institute for his research on plant sciences, where he worked on molecular characterization of abiotic stress, inducible genes and signaling response, with rice as a model system, and received his PhD degree from Jabalpur University. During his postdoctoral work, his chief focus was on developing salt and drought-tolerant transgenic rice. Dr. Choudhury is currently handling several government-funded projects on abiotic stress responses in rice and supervising several PhD students as the principal investigator at his institute. He has over 200 years of research experience in the field of environmental stress tolerance mechanisms in plants. To date, he has published over 150 articles in peer-reviewed journals and many book chapters in several reputed international and national publishers. He has edited several books from well-known publishers like Wiley, Springer, and Elsevier, and he is a life member of different scientific associations and societies, as well as the recipient of the Young Scientists Award in 2019, awarded to him by the International Foundation for Environment and Ecology at the University of Allahabad, India. We are highly honored to have him as our guest speaker for today's webinar. It is now my request to all of our viewers to enjoy the talk and to hear Dr. Choudhury speak. Whenever you're ready, sir, we would love to hear from you.
Okay, thank you, Liberty. Thank you, Bioingene, and a very good morning to all of you. So, shall I start now?
Yes, yes.
Oh, okay. Okay, thanks to the entire Bioingene platform, and I really appreciate the endeavor by Shubho and his team for framing such a wonderful scientific platform. You know, it gives so many people the chance and the opportunity to get together, because in our research career or teaching career, we hardly find time to make out precious time to indulge ourselves in these kind of endeavors. So it's really a very I should say a very appreciating kind of endeavor by Shubho and his team. So once again I congratulate him for this nice effort. Okay, so the topic that I have chosen today is journey of rice research from salinity to fluoride. So what I would be trying to highlight is some of my works based on rice research, because basically I know very little about other crops. Okay, I know a very little also on rice, but that also for other crops I know nothing almost. So I try to share some of my experiences with rice research. So let me share the slides. Uh, okay, so is it visible?
Yes, it's good.
Yes. Okay.
Um,
Okay. So I continue. So first of all, salinity stress. I will try to yes.
I can see the speaker. I mean, is it? I mean, Shubha, is it my view problem or everybody can see the?
No, it's your yeah. It's
Okay, okay,
okay.
okay.
You
Okay. Okay.
Thank you.
Okay. Shall I proceed?
Yes.
Shall I? Okay. Okay. So I have tried to actually dissect the entire discussion into two parts. So first part, because salinity stress is one of the areas in which I used to work during my PhD postdocs. And then one of my PhD scholars, he also worked on salinity stress, and thereafter I shifted to fluoride stress. I will tell you the reason why I did it. So first of all, I would like to talk much on salinity stress response, and then the later parts of my work, that is which I am currently doing, that is on fluoride stress. So what is meant by abiotic stress? Basically, when you talk of plant science, okay, some of the thrust areas of plant science, particularly with respect to rice research, okay, are number one is nutrition. Okay, nutritional profiling and nutritional enrichment, which we call as biofortification, which I'm sure Shubho is also working probably, and also other members. Many of the research teams in India are working on nutrition. And the other one is the stress. The chief two areas. Stress means it can be again demarcated into two groups, basically broadly, that is biotic stress and abiotic stress. By biotic stress, we mean mostly the pathogen infection. It can be bacterial, viral, it can be fungal. And the area in which I am more concerned is the environmental stress. And when you talk about abiotic stress, these are the different, these are the different areas: osmotic stress, the water deficit, drought, salinity, the heavy metal stress, the extreme temperature, high temperature, or chilling. okay, of which I am more focused with drought and salinity, mostly salinity, and also to some extent drought stress, and also the heavy metal stress. I don't work much on this extreme temperature, high temperature or chilling, because chilling is not a problem in India. Of course, high temperature is a problem, but chilling is not a problem in this part of the world. Now, the problem with abiotic stress is that, unlike biotic stress, the problem with abiotic stress research, one of the problems I would say is it's characterized by multigenic trait. That means it is very much, you really need to be very fortunate that you just clone one single gene and you get a desired response, because that single gene is going to have an elaborate and extensive crosstalk with so many pathways, as you can see from this figure. That even that the single salt stress, when you impose a salt stress, it on one hand it is triggering the gene activation, osmoprotectant, chaperone, detoxification, and transporters. Okay. On the other hand, you see from drought also you are getting the same response. Okay, so there is a crisscross, or there is a crosstalk between the different stresses. And you have to remember that in an agricultural field, okay, when the plant is actually within nature, there are so many simultaneous stresses acting upon it. Okay, it is not only when you are working in a lab, you are focused on salt, you are focused on drought, but when you are in the actual field, the same plant can be challenged with so many stresses. And so this crosstalk, okay, is very much important. And what is governing the stress tolerance is actually through different cascades, signaling cascades. And you have a series of stress associated genes and proteins, which leads to the acquired plant stress tolerance. In pathogen infection, we call it systemic acquired resistance. Here we just designate it as acquired stress tolerance. Then it ultimately leads to the induction of different stress related metabolites, the different antioxidants, the different transporter proteins. So you see that one single gene, if you incorporate, it does so many things in order to generate the stress tolerance. So this was one of our chapters which we actually showed in this particular chapter, that there is a global expression analysis of abiotic stress responses, and there is a distinct clear-cut difference between the sensitive and tolerant plants. With respect to rice, I will be focusing on this because this is the area in which I have been working for a long time. And then you need to fish out all those genes and proteins through different technologies like microarray. You have the microarray, the multi-top, the Is2 hybrid systems. Okay, and finally you can go for the genetic enhancement, that is through transgenics. Okay, I will talk about the transgenics, the pros and cons of transgenics, why the genetic modification should be given more and more emphasis without being criticizing too much about it. So I will come to that in the later part, okay, particularly during the question answer session or during the interview session. Now, why rice? The question is why rice? Number one is, you know, that in this part of the world, Southeast Asian countries, rice is our staple food, and West Bengal, if you even compare West Bengal, in which I am working, it's known as the rice bowl of India. Okay, so so it feeds most of the Southeast Asian countries, and West Bengal is one of the leading producers of rice also. And this rice, it exhibits a great varietal difference. Okay, because the same Oryza sativa rice, it has certain salt tolerant varieties and as well as certain salt sensitive varieties, of which the tolerant varieties are mostly the Pokali and Nonabokra, and the sensitive varieties, there are other varieties also, but these are the varieties in which I am working for quite a long time. And the sensitive varieties like M148, IR29, IR64, these are all sensitive varieties. And one of the major phytohormone which governs stress tolerance is the endogenous ABA level. And it has been found, this is a well-known report, that the tolerant varieties, they have a higher endogenous ABA level than the sensitive varieties. Now, whenever you impose any plant to salinity, now I strictly restrict myself to salinity without going too much on other stresses, because that is my main focus. So when you expose a particular plant, even rice plant, it has, as I told you, it triggers different oxidative damages, and the plant's physiological means of fighting against those damages is through triggering of different osmolytes, antioxidants, and so on. And along with that, there are several ABA inducible genes. That means the endogenous ABA level is activated, and simultaneously what happens is it triggers different ABA inducible genes, of which the major group, one of the major groups of genes is the LIA, or the late embryogenesis abundant genes. And of that LIA, the gene in which I have been working for quite a long time is the RAB, which means responsive to abscisic acid. And there are several groups of LIA. I'm not going into that, but this RAB is actually group 2 LIA gene, or it belongs to the dehydrin family of proteins, whose main task is to ensure some kind of protection. But how RAB actually confers that protection, okay, that was first deciphered actually through our work. Okay, because previously people did not go for that well characterization. It was a reported gene, one of the genes which was reported quite early by Mundy and Chua, John Mundy and Namhai Chua, but the gene was not characterized. Okay, so that was one of my earlier works, you can say. And we also characterized the promoter of this particular gene. Initially, it was shown that this promoter, it has actually two motifs. One is called motif 1 and another is called motif 2. And motif 1 is a typical ABRE. What is meant by ABRE? That means a promoter sequence with having an ACGT code, which can sense the ABA, which can sense this increase in or enhancement in this ABA level. And also the motif 2, the function of motif 2 with respect to RAB16A was not characterized at that time. And so we actually, when during my early PhD days, we tried to actually focus, rather my supervisor along with me, we tried to focus on the characterization of the promoter sequences. Because what is the transcription factor that binds to this promoter sequence? That was not very much well characterized. Okay, so I think it's clear to you what will be my main focus. Okay, so with that lacuna, we tried to first study the gene map of RAB16A gene. As you can see that this is the promoter sequence with Tata and Cat box. These are the motif 2 and 2, motif 2A and 2B. I mean, this is probably one of the sequences which is present along with motif 1, which is the ABRE having this ACGT code, of which the function of this motif 2 was not characterized at that time. Okay, and what is the transcription factor, as I said, that binds to, that was also not well decided. Now, when you compare this RAB16A gene expression among sensitive and tolerant variety, as I told you, this is a sensitive and this is a tolerant variety, and we also chose one aromatic rice, because I have been working on aromatic rice also for quite a long time. So when we compared it, you can see that when you apply stress, okay, you see the induction, and even at the control condition, you see the RAB16A gene expression, but you don't find the expression in the sensitive varieties. Which means that a very good observation that in case of the tolerant varieties, you have these genes which are present even in the control condition. That means they are already predisposed or they are already pre-induced to stress, because they live in such an environment, okay, where you find the gene expression even under control condition. And if you apply stress, it is slightly triggered. Okay, so this was one of our observations, that there is a clear cut, you can say that there is a clear cut difference, varietal difference between the sensitive and the tolerant variety. Okay, now initially we tried to characterize whether the RAB16A gene, it really, so because the expression is higher in the tolerant variety, so it gives us an impression that probably this gene will confer some sort of tolerance. Now, before going actually into the rice transformation, what we did was we chose a model plant, which in plant system is either Nicotiana tabacum, tobacco, or Arabidopsis. So what we tried to show is whether this gene confers salt tolerance in transgenic tobacco. But before that, we have to confirm that whether there is any nuclear factor in tobacco that actually binds to Rab16A promoter. So what we did was we just performed a gel mobility shift assay or electrophoretic mobility shift assay with the nuclear extract from tobacco, and we showed that yes, there is a binding, and this binding is lost when you apply the competitors, which shows that yes, this binding is very much specific. So that means probably there is a factor, transcription factor in tobacco that can recognize Rab16A, so that it will be quite worthy if I can go for overexpression of Rab16. So we cloned it in the, these are some of the routine works. We cloned it in a plant expression vector PBI121. We generated the transgenics and confirmed the transgenics through genomic PCR and southern hybridization. Then we expose the transgenic plants. These are the different transgenic lines, and expose them to NaCl, AB, and PEG. This PEG means this actually induces drought. PEG actually induces, or it mimics the drought stress. So you can see that the Rab16A gene is being induced. Okay, and even when you do Western blot, you find that in different transgenic lines you are getting the expression, protein accumulation. So when you perform a salt tolerance assay in comparison to the wild type plant, if you go for the transgenics, you can see that at least this transgenic plant, it behaves quite nicely. Okay, it grows quite happily even under 300 millimolar NaCl. And so far as vegetative and reproductive growth is concerned, there is not much difference between the wild type and the transgenic plants. When you go for the different measurement of different agronomic traits, if you see the wild type plant, there are no flowering or no seeds under 200 millimolar NaCl, whereas if you see in case of transgenic plants, okay, they are able to flower and they are able to produce seeds. I will not go into details of all this because these are some of the routine physiological analysis. I mean, whether the plants, they are performing well with physiologically, transgenic plants. So to show that, we perform different biochemical analysis. For instance, the growth inhibition, the total chlorophyll content, the MDA content, malondialdehyde. This is an index for lipid peroxidation. Okay, then we measured the sodium and potassium level, because you know the Na plus K plus ratio, or rather the K plus Na plus ratio, that is very important, because if a plant is tolerant, then obviously the sodium level will be lesser, because sodium is a toxic ion. So potassium level will be higher in this case. So those kind of analysis were done. We measured the different osmolytes, particularly the proline, the total polyamines, the different antioxidants, the polyamines. I will come to polyamines later. The putrescine, spermidine, and spermine. Polyamines are actually, they also confer protection. They are kind of osmolytes. So all these analyses were done. The different antioxidant and antioxidative enzyme activities, these were performed, and we published the paper in Plant Cell Reports. This was way back, of course. I told you that these are some of my earlier works. And then when I worked with Professor Shapon Datto and Madam Dr. Korbi Datto, then we actually went a step forward and tried to overexpress this Rab16A gene in rice. And in that case, in this case, what we did was we chose one of the sensitive varieties, that is Khitish, okay, which is quite sensitive to salt stress, and we use the particle bombardment technique to transform the rice plants. So in a similar fashion, what we did was we confirmed the presence of the transgene through genomic PCR, and showed that it was behaving almost normally like the wild type plants, like the control plants. The transgenic plants were checked or confirmed through Western blot analysis, where they are showing the protein accumulation in presence of stress, and also through the the semi-quantitative RT-PCR, where they are showing that in case of NaCl stress, there is a 8.6 to 18.2 fold induction among the different transgenic lines. There are induction of this gene, and you can see that when you compare against wild type and transgenic plants, you can see in presence of stress, the performance is quite better. And even when you measure the agronomic traits like plant height, the fertile tillers, the spikelet fertility, you can see the transgenic plants are behaving better. So that was published in Plant Signaling and Behavior. And these are some of the biochemical parameters that were tested. Overall, the conclusion was that the plants were behaving better. And this year we also published this paper where we are showing that the OS RAP16A gene is also showing drought tolerance. Okay, so you just imagine the work that I started during my PhD, it culminated this year. Okay, if I would at all regard it as culmination, then it actually ended this year. So you see how much time it takes for a well characterization of a particular gene. Okay, so you can see here we took one aromatic rice variety, transgenic aromatic. This is Pusha Sugandhi 2. And so here also we showed that if you compare against the wild type plant, you see the performance. Okay, so the overall with both with DREB as well as DREB is actually a transcription factor. it is also of course not connected with ABA dependent pathway. It is connected with ABA independent pathway, because in abiotic stress, okay, there are several genes which are also induced in an ABA independent fashion. Though my focus was mostly on ABA dependent, but still we actually made a comparison between an ABA dependent gene and an ABA independent gene, and we showed that yes, both are equally capable of generating the drought tolerance. Okay, because if you withhold water for 21 days and impose them to drought, you see that still the plants are quite healthy. So this is one of our recent papers. And now the next part of the work is, not next part, the sub part, I would say. Now, what is the transcription factor, okay, that binds to the RAP16A promoter? This is the next question. So we, with regard to that, we actually published two papers. One is in BMC Plant Biology and another is Plant Cell Reports, where we are showing this OSBZ. Okay, this is one of the transcription factors that was reported in rice, and it was actually shown to target a large number of genes, okay, in context to the AB inducible expression. But whether OSBZ8 is also a master regulator for RAP16A, it was not known at that time. So what we did was, because I'm working on rice, one of the advantages of working with rice is that the genome sequence is known. So you can very easily, you can try to what you say, you can clone a gene very easily. Okay, so what we did was we cloned the OSBZ8 cDNA in a bacterial expression vector, which is PR-SETE, and we purified the protein. That is through this is the 6X histidine tagged OSBZ8 protein. We purified the protein, and then we did again an EMSA. Now this time we used two separate experiments. One is we prepared rice nuclear extract using again a sensitive variety and a tolerant variety. Okay, see the difference. In case of sensitive variety, if you have no stress control, you are not getting any expression, which is quite according to expectation. In stress, you are getting only a feeble expression, very weak induction. In case of control and stress, you see in the tolerant variety, you are getting quite high expression. And when you use competitors, you are again, the complex formation is abolished. But the interesting part which we got is that here you are getting two complexes. You see, you can get two bands. One is lower one and another is the upper one. So initially we were quite confused why we are getting these two kind of complexes instead of just one. Okay, now what we did was to further check whether the OSBZ8 cDNA is over here or not. What we did was we used an antibody against OSBZ8 in the EMSA experiment. So you can see the complex formation is abolished. Okay, so which means that something is happening. This complex is not there. Something is happening, because if you apply the post-immune sera, you see that the complex is not there. But still, we are not. We probably made some sort of hypothesis that yes, it can be OSBZ8. But what we did to further prove it, when you apply directly the recombinant OSBZ8 and see the binding, check the binding with RAP16A promoter, you see you are getting a huge complex. And that complex actually it migrates or it tallies with the lower complex, but not the upper complex. So this gave us a conclusion that one of the transcription factors is OSBZ8, but there is yet another factor. And unfortunately, this yet unidentified factor remains unidentified till today, because of the reasons that somehow I left the lab, and somehow this work was not, it was not continued. But I would love to someday again dig out this work and try to find out what this complex is. Okay. Similarly, if you undergo a southwestern blot, you can again see that there is a two complex. One is 38 kilodalton protein and another is the 29 kilodalton protein. Now we are sure that the other protein is probably 29 kilodalton. That we are quite sure. Okay. And even when you elute the protein from this complex and go for a silver staining, okay, if you now, okay, I missed out one thing here. What we did was we used motif 1 and motif 2 separately. Okay, because I told you that the RAP16A, it has two motifs. One is ABRE motif 1 and another is motif 2A, which is a GC-rich sequence. And probably we hypothesize that it behaves as a coupling element, which works in conjunction with the ABRE sequences. Okay, now when you separate them, and if you check the binding with the nuclear extract, you see that here the binding is stronger. That means with the typical ABREs, the binding is stronger, but with the GC-rich sequence, you find that there is a weaker induction. Okay, now what we did was we just eluted this. Okay, we eluted the protein out of this complex and we silver stained it. Now from this complex, we could get only a 38 kilodalton protein, whereas when you elute this motif 2A, you are getting two proteins. One is 38 kilodalton and another is 29 kilodalton. So which actually matches with this. So probably this 29 kilodalton protein is recognized not by motif 1, but it is recognized by motif 2. Whereas 38 kilodalton protein is recognized both by motif 1 as well as motif 2. So probably it is a heteromeric complex. Okay, heteromeric complex between 38 kilodalton transcription factor, which is OSBZ8, and 29 kilodalton protein, which is yet unidentified. So we published that. Actually this much work we published it. So this is actually, I'm not repeating, this is actually the same experiments where we showed binding with recombinant OSBZ8 separately with motif 1 and motif 2. Another interesting observation that we found was this binding is actually facilitated by phosphorylation, because you know that in case of salinity stress, okay, the much of the signaling cascade, it depends on the presence of different calcium dependent kinases, and there is actually a calcium burst during salt stress. Somehow the calcium channels, they get opened, and the cytoplasmic calcium concentration gets increased, and thereby the different kinases, they get activated, most of which are calcium dependent. So we asked the question that whether the calcium or whether the kinases or phosphorylation, they have any role in this binding. So when you apply ATP, okay, or when you apply casein kinase 2, mind that casein kinase 2 is one of the proteins which is also, it activates RAP16A gene or any RAP gene, casein kinase 2. So if you use this casein kinase 2, or if you use this, the ATP, okay, the induction or the binding intensity gets increased. But if you apply heparin, which is a competitive inhibitor, you can see that the binding is abolished. And when we apply SAT fraction is nothing but it's a cytosolic extract which is presumed to contain the kinases. So when you apply this SAT fraction also to the gel shift assay or the electrophoretic mobility shift assay, you see that there is an induction of binding. So all these conclusions, all these observations led to the conclusion that this binding is facilitated, or this is further intensified by phosphorylation mechanism. So we ended the work over there, and then we went a step forward again with where we actually characterized certain promoters. The RAP promoter, the 4x ABRE promoter, that means having 4 tandem repeats of ABRE, the 2x ABRC where the ABRE and the coupling elements are present together. And this was shown in, of course, in rice again, the promoter characterization or the promoter inducibility. So this is the colorimetric gas analysis where we showed that the 2x ABRC promoter is probably the strongest, because when you apply 150 millimolar NaCl or 100 micromolar ABA, you see the strongest induction of gas, it occurs in 2x ABRC followed by 4x ABRE, and RAP promoter is rather a weak promoter because it is having only one ABRE. Whereas here you are using ABRE and ABRC, 2 repeats. Here you are using 4 tandem repeats. So this gives us the probability, or this gives us the scope, that if you can use these promoters for induction of any gene during salinity stress, you can get desirable results. Okay, if you use 4x ABRE or if you use 2x ABRC, okay, and clone any gene downstream of it and then go for the induction of any gene and generate salt tolerance, then you can get much better response than using RAP promoter, which is comparatively weaker promoter. It's a natural promoter. Okay, so in a similar fashion, we also went for the DRE based promoter. I'm not going into details because this is mostly the dehydration responsive element promoter, which is not induced by ABA, but it responses to NaCl and PEG or even cold stress. Okay. So we published those papers. These are some of the review papers based on these works, Lia protein and basic transcription factors. This was one of the works of my PhD scholar, where he characterized several agronomic traits. And this was published in JPGR, Journal of Plant Growth Regulation, where he measured the different panicle length, the rachis length. So this is purely, the agronomy based work. And then we also tried to construct a heat map based on the characterization of some of the very well-known genes, the transcription factor, the osmolyte biosynthetic genes. Okay, and these are some of the growth-related genes, grain yield-related genes. Okay, so these were actually constructed, and some of the conclusions that we got was, in case of the tolerant variety, there was lower reduction of grain weight and field grain number. Okay, in Nonabokra as compared to the sensitive variety, that is IR64. Now, the another part is regarding, as I already told you, that this has also been one of my very one of the areas which interests me, that is the aromatic rice, because in India, and you see there is a diversity of scented rices, and the main compound that is responsible for this aroma is known as the 2-acetyl-1-pyrroline. And also, although there are several other compounds as well, there are different other volatile compounds, but that is the major compound. And in West Bengal, we have this popular aromatic rice called Govindabhog. I already showed some of the earlier data that the expression of RAP16A gene in Govindabhog. And in addition, there are also several other indigenous rice cultivars, indigenous aromatic rice cultivars, okay, whose aroma formation, characterization or pathway is not known, number one. And number two, how they respond to the salinity stress, because they are very profitable. They have a very important economic trait, okay, and that is the high aroma. So it is very important that we should know how the stress tolerant pathways or how the stress susceptible pathways operate in these varieties. So with that aim in mind, I tried to find out some of the biochemical and molecular features in this particular aromatic rice variety. So initially, this was one of the works, and this was actually covered by one of the leading, what should I say, scientific magazines, Deccan Herald, who actually made a coverage. The reporter came and they actually made a coverage of our work in Govindabhog. And for the first time, we showed that Govindabhog aromatic rice is actually highly salt susceptible. Okay, and even it is much susceptible than the sensitive varieties. Okay, because it's a severe kind of damage. So these were some of the traits that were measured, and I'm not going into details. I just told you the summary of this work that we did. And then this was Pooja. She is working in my CSR project on aromatic rice. And there we showed, we characterized as many as 11 to 15 indigenous aromatic rice varieties, and we showed a commonality in the pathway regarding the aroma formation. Now the interesting part is that the metabolites which are connected with stress tolerance, for instance proline, for instance glutamic acid, for instance the polyamines, putrescine, spermidine, spermine, GABA, the gamma beta, the gamma aminobutyric acid. Okay, so all these metabolites are also very common metabolites for, you know, the salt tolerance. But at the same time, these are also involved in aroma formation. Okay, so this was the pathway that we characterized in case of indigenous aromatic rice varieties regarding the aroma formation. Now here, please pay attention here regarding 6A and 6B, which is a very important enzyme called the betaine aldehyde dehydrogenase 2. And the status of betaine aldehyde dehydrogenase 2 actually decides how much aroma formation will occur. Okay, because you see that in 6A, this is a non-functional betaine aldehyde dehydrogenase 2, because if betaine aldehyde dehydrogenase 2 is mutated, or if it is inactive, or if it is truncated, then this gabalt will go towards delta 1 pyrroline formation and aroma formation. But if the this enzyme is active, then what would be your fate? This will not move towards this pathway, this will move towards the GABA pathway, and so there will be less aroma formation. So one of your strategies in transgenics could be, if you could downregulate, or if you can go for RNA interference of bad H2. So in that case, you can, that will give you a probability of increasing the aroma formation further in the aromatic rice varieties. Okay, so that is a very important observation. Anyway, my interest is more on what happens when these aromatic rice varieties are challenged with stress, because at that time these metabolites will be confused. What we will do, we will go towards aroma formation, or we will save the plant, because you know survival is one of the first priorities in any living system, whether a plant or an animal. So at that time, the plant will be confused whether I will follow this or I will channelize this metabolites towards stress tolerance. So what we did was, we chose, actually this was published in Plant Growth Regulation, where we chose actually 4 different indigenous aromatic rice varieties: Tulai Panji, Radhuni Pagol, Kalonunia, and Govindubhog. And we compared them, these 4 varieties, against one non-aromatic variety, which is IR64. Now I will just give you the summary of this work, because this is already a published work. One can go through the papers. The findings that we got in this paper is that, except Calonunia, this is an exception. Otherwise, all these 3 varieties, they are channelizing, even during stress, they are channelizing the metabolites towards aroma formation. They are least bothered about stress tolerance. But Calonunia, in case of Calonunia, what happens is that, okay, these metabolites are more channelized towards stress protection, because you can see that in Calonunia what happens is that the bad H2 is higher, betaine aldehyde dehydrogenase 2 is higher. If betaine aldehyde dehydrogenase 2 expression is higher, which is corroborated through Western blot, then that means that, okay, you are getting lesser aroma, right? You are getting more, they need the metabolites more for their protection. So this was one of our observation. Of course, it needs to be checked out with other aromatic rice varieties also, but preliminarily we have just selected, because these are the major, you know, these are the major aromatic rice varieties which are known to have pronounced aroma, very high aroma content, and so they are economically very much, you know, they are economically very important in that respect. So we chose initially these 4 varieties. So these were some of our conclusions. And we also, I'm not going into details of this work, because here we analyze the different antioxidant and nutritional profile in the unpolished and polished grains of different aromatic. This is not connected with salinity stress, so I'm not going into details of this. Okay, this was one of the works with the polyamine biosynthesis. I think I will skip this one, and I will come to next part, that is the fluoride stress response. Okay, so here again we why now the question is why I have shifted to fluoride stress. The obvious answer behind this is that if you see the present scenario, okay, if you see 1999 versus 2018 report, you can see the distribution of fluoride in groundwater in India. It is gradually increasing. Not only in India, I have focused because I'm working in India, so I have focused the map of India, but it's a global problem. It is gradually turning out to be a global problem. And in India, there are hardly any state, for instance, only this part which is marked with green maybe. Otherwise, um, uh, fewer or higher, almost every state of India is now affected with fluoride. Okay, and see, in 1999 the scenario was like this, and now the scenario, it is gradually going getting more and more increased. And it is said that if you surpass 1.5 mg per liter level of fluoride, then it's becoming a poison, or it's a poison. And already in Indian states you have this much level, that is 48 mg per liter. Now again, why I chose rice? Because groundwater contamination, and you know, rice is it's one of the water intensive crops. It needs a lot of irrigation water for its for its productivity or for its growth. So obviously, if your water is contaminated, then you are actually feeding rice plants with polluted water, and automatically this will be uptaken along with the conduction system. And in case of rice, when I started this work, this is one of the works which is still continuing under my ACRB project. The mechanism of fluoride toxicity in rice is completely unknown. It has been, there are some fragmentary data with other crops like Triticum aestivum or Brassica, but that also very poor data. I mean, it's not elaborate data. Okay, only few, maybe some of the assays have been done. So that made me interested in this kind of stress. So initially what I have started, this work has been started only in 2018, because prior to that I was mostly focusing on the salinity and drought stress response. So here what we did was, we initially characterized, I will just summarize the overall work without going into too much of details. Now overall conclusion I would be just talking about. So here what we did was, we chose one IR64 and another one aromatic rice, again that is Govindabhog. And initially we showed the toxicity level of fluoride toxicity and the different defense mechanisms, okay, in response to fluoride toxicity, because nothing is actually known about these type of mechanisms. So you can see this figure, that when you place two plants side by side, you see that in IR64 the damage is much more intense as compared to the aromatic rice. So here aromatic rice, unlike salt stress, is not behaving that much susceptible. Okay, it is behaving some sort of, it is showing some sort of intrinsic tolerance. So see the beauty, the same Govindabhog which was showing extreme susceptibility to salt, okay, is not behaving in a similar fashion, okay, with respect to fluoride stress. Okay, so you may not be knowing from outside that the plant is facing fluoride stress. It is going very happily, but what is happening inside, okay, that is more disastrous. Okay, so that was one of our conclusion. I will not go into details. We estimated all the fluoride contents and the different physiological parameters, the different expression of different genes, okay, which are governing the stress tolerance, the proton ATPase, which is one of the membrane proteins. We check the expression of these proteins in response to fluoride stress. So I will just skip and go to the conclusion that we showed that one of the chloride channels, CLC2, okay, it appears to be involved in fluoride uptake. This was one of the conclusion. And as I already told you, IR64 is behaving as a susceptible variety, whereas Govindabhog is appearing to some extent as a tolerant variety. These were some of the defense mechanisms, like the osmolytes, polyamines, the different antioxidative enzymes. And I just want to draw your attention towards this abscisic acid content. Okay, normally in case of, as I talked about salinity, that there is an induction in the ABLL during stress. But see here, when you check IR64 in comparison to control, when you apply stress, there is a decrease in the ABLL. Whereas in case of this Govindabhog, you see that there is an induction in ABA level. So you see, in case of fluoride stress, ABA is behaving in an erratic manner, or in a quite abnormal fashion. Okay, so I will not go into details of ABA action because my paper is yet to come on that. So I will not be discussing in details about it, and I will be able to comment on its position only after the paper is published. But this is a very interesting observation that we got. Okay, and so the different antioxidative enzymes, the expression of different antioxidative enzymes, the osmolyte biosynthetic genes, so these were all characterized. Okay, the isozyme profiling, these are routine biochemical and physiological analysis. Then the different genes which are connected with ABA biosynthesis, particularly NCD3, the ABA ATOX that is actually concerned with the ABA degradation, the different ABA inducible genes, the expression of ABA inducible genes and transcription factors, these were analyzed. Okay, and so this is one of the important conclusion. GB accumulated, GB means Govindabhog accumulated higher ABA level, IR64 having slow ABA degradation. So as I told you that ABA is behaving in a contrasting fashion in the two cultivars. Now, whether we could use certain protective agents to overcome this fluoride stress. See, when you confer protection in the first part of my work, okay, when I showed you, we use the transgenic technology, whether in tobacco or in rice. But here initially, okay, before going into transgenic, because it will need a little bit more time for me to characterize few genes, okay, with respect to fluoride stress, because I know nothing is known about it. Okay, so it will probably take time. Till that time, is there any other sustainable approach, or is there any other cost-effective approach to overcome this fluoride stress? So one of the approaches which people follow is either you apply it exogenously in the form of foliar spray, or you can go for the seed priming. That means you pre-treat the seeds to some protective agents and then expose the seeds to the stress condition. So we did exactly that, and I started with one of my very favorite items, you can say, protective agent, that is polyamines. That is initially here, that is these are again all published works. I'm showing actually all my published works basically. So I'm just going through the summary that we published this in, where we showed that spermidine, okay, it has a very potential role in overcoming fluoride stress, because you can see here, okay, when you apply 0.5 millimolar spermidine, you are getting the induction. Okay, and if you see here the fluoride content, then also the fluoride content here is comparatively lower down in presence of polyamines. Okay, Shubho, shall I get another 10 minutes?
Hello.
Hello, can I get
Yes.
another 10 minutes?
Yes, sure.
Okay,
Sure,
okay,
sure.
okay. So 0.5 millimolar spermidine, as you can see. Again, these are some of the routine works. I will not go through this. It's again the same, that measurement of the different parameters, the measurement of the checking the expression of different genes, different physiological issues like sugar metabolism, nitrogen assimilation, whether these are being improved or not as a result of spermidine treatment. So all these have been checked. And you can see here also, this is again interesting with respect to ABA. See, with fluoride, there is a lowering of the ABA level, but when you apply spermidine, again the ABA induction occurs. So ABA has been measured, you know, that there are the sigma actually provides the ABA immunoassay kit. It's a very sensitive kind of kit which you can use for this kind of work. So ABA accumulation has been measured. Again, some of the crucial genes, Rubisco, we all know that it's a major photosynthetic gene. So the fold changes of Rubisco, the spermidine, it actually improves the Rubisco expression, gene expression. So all these parameters were actually measured. Okay. So these were some of the conclusions, that exogenous spermidine, it actually reduces fluoride bioaccumulation. Now, while spermidine is a triamine, there is another polyamine, higher polyamine, which is tetramine, which is called the spermine. So in a similar fashion, we also showed that spermine, it confers protection against fluoride toxicity, and this has also been published in ecotoxicology, environmental safety. So you can see here that the performance of the plant. So again, these also have been analyzed through different tests and through different molecular analysis. I'm not going into details. Now, calcium. This was also one of my one of the papers of my PhD fellow Ankur. So where he is actually showing that if you prime the seeds, priming means if you are pre-treating the seeds with different calcium compounds, for instance, calcium hydroxide, calcium nitrate, or calcium chloride. So you can see that in comparison to sodium fluoride stress, you can see that the performance is much improved. So what is again the molecular mechanism? Now what calcium actually does here is calcium actually tries to chelate. Calcium has a tendency where it can actually bind to the fluoride and it can precipitate it out as calcium fluoride, CaF. So therefore the overall effect of fluoride, okay, in the plant system, it gets much reduced. And surprisingly, in calcium hydroxide and calcium nitrate, if you apply 0.3 millimolar, they are behaving better. But in calcium chloride, you see 0.3 millimolar is not behaving in that much better. See, 0.5 millimolar is working better. Okay, so it is very much concentration dependent. The concentration of these protective agents are very important. Okay, in conferring the protection. Okay, so in a similar fashion, we also showed melatonin. Now I will just spend a few time with melatonin because melatonin because most of us know that melatonin is actually an animal, it's something, an animal metabolite. But the plants do have melatonin, and particularly in rice, you have quite a significant amount of melatonin. And in plants, apart from circadian rhythm, regulating circadian rhythm, melatonin, a lot of work is going on in recent times where they are showing that melatonin acts as an antioxidant. So we asked the question that whether this melatonin is equally capable of generating fluoride, reducing fluoride toxicity in rice seedlings. Okay, because melatonin and abscisic acid, they are acting antagonistic to each other. That is reported. So if you remember, our experiment showed that there is a lowering of ABA level. So that means we asked the question that is it because it is more guided by melatonin pathway rather than ABA pathway? And we found that yes, it is really so, because when you apply melatonin, okay, or when you apply gibberellic acid, both are actually antagonistic to ABA, right? And both of them are giving quite pronounced response. So in this fluoride stress, ABA is not probably ABA is not that important. Okay, rather than that, these are more important regulators, because melatonin, if we find that it is negatively regulating the expression of ABA dependent genes. Okay, and so in probably the fluoride stress is more governed by melatonin and GA pathway rather than ABA pathway. So this was one of our from this kind of work, if you combine all these data, then that is one of our important observation. Similarly, we used silicon because rice also has a large amount of silicon content. If you go for the reproductive phase, so long phase, now what we were fortunate to have 3 different varieties with 3 different patterns. We chose IR64 and GB like the previous experiment, and here we are again using Khitish, because Khitish again is one of the varieties that I was using quite a long time back. when I used to work with Professor Dotto, we actually generated transgenic khitish. So now I was really fortunate to choose these 3 varieties, because if you check the grain development, okay, you see that
The
IR
screen sharing is not working, it seems. Could you please
Hello.
check?
Sorry. Yes.
hello, can you hear me?
Yes,
Hello.
audio is good.
Sorry. Yes. Are you
Okay, just
in?
is it visible now?
Not yet.
Not yet. Okay. Okay.
Yes, it's visible now.
Visible now? Okay.
Yes.
Okay. Okay.
Yes.
So if you just go check the pattern of grain development, you can see that it's very much suspended over here. It's moderately suspended, but it is growing quite happily in case of khitish. So probably we now have a tolerant variety, a semi-tolerant variety, and a susceptible variety. So you have 3 different types of grades of the tolerance or susceptibility pattern. Okay, again I am not going into details. We measured the different the Ross accumulation. We showed that it was maximum in the grains of IR64, followed by Govindobod, and least in khitish. So khitish is behaving more as a tolerant varieties in this case. Okay. So this was one of our observations. This work has been covered by the East Coast Science News. This is only in September 18, 2020 that they have made a coverage of our work on fluoride accumulation. Okay, so it's an internationally accepted data now. And we used nanoparticle. I've just started working with nanoparticle, where we have actually generated silicon nanoparticle, and we have shown that it mitigates fluoride stress in rice. So these are actually the silicon nanoparticles generated. Okay, they are giving good kind of protection to the rice plants. So these are some of the work. Again, I'm not going into details. Now, another interesting thing is that we have now tried to go for a de novo RNA-seq analysis to find out which are the genes, because otherwise we cannot go for the transgenic approach. So this was done, and the conclusion of this work is that we could actually find that there are almost 7745 transcripts, 2698 transcripts, and 5 transcripts which we could retrieve from the different databases. And again, we chose some of these genes, and we made a comparative analysis between the sensitive and tolerant variety, which in this case is IR64 and khitish. And we showed that some of the genes which belong to these families, they are very important, like the cytoskeletal remodeling, altered xenobiotics, cellular autophagy, nucleosomal remodeling, inhibited abscisic acid, activated gibberellic acid. So now we can actually try to find out which are those genes among each of these groups that can confer considerable fluoride tolerance or fluoride protection. So we are now having quite a large number of genes in our hand. Okay, and so that we can go for those kind of transgenic approach to generate fluoride toxicity. Okay, now this is work. You see that if a plant is being challenged by arsenic and fluoride together, because arsenic contamination also comes from groundwater. So if by chance there is a simultaneous co-contamination, then a tolerant variety like Muktustri, see what is the condition. Okay, see the condition when if a plant it uptakes both arsenic and fluoride together, then one stimulates the transport of each other. So this level also enhances, fluoride level also enhances the arsenic. So see the dangerous situation of the plant, and the rice as an item we are consuming that means we are actually, it's a biohazard, we are consuming that. So we should be very careful, and something needs to be done in order to address this problem. Okay, so that is all about it. Simultaneously, we also showed lead fluoride and nickel fluoride co-contamination, but here lead fluoride is behaving like arsenic fluoride, but nickel fluoride is behaving in a different manner. The summary of the work, because nickel is actually trying to protect against fluoride stress. Okay, nickel is also a kind of, it's not a very pleasant metal, but even then nickel is giving protection. Whereas if there is a lead fluoride co-contamination, then there is it's a severe kind of toxic situation like the arsenic fluoride. So you see the combination of the metal and fluoride, that is also very much important. Not always there will be equal danger. Okay, sometimes there may be a promoting effect also, sometimes there may be a protective effect also. So that was actually shown through this particular work. We are also undergoing melatonin response in arsenic stress. I'm not going into details of this work. So these are actually my fellows. And Aditya is actually the main pillar of my work. Ankur is also working on fluoride stress. Shantanu working on arsenic stress with respect to melatonin. Pooja on aromatic rice, mostly in my CSR project. So that is all about it. I hope I have not bored you or talked too much. Okay, so thank you for giving me this opportunity once more. Okay, so thank you. So I stop sharing. Okay, I stop sharing my slides. Okay.
Yes, and I
You're not, your
already
video is, yeah, now it's okay. All right.
Yes, it's boring at a time.
Yeah, Shubho's internet seems to be having some sort of problems.
This
All right.
here I have already highlighted. So your talk is very interesting, and it's because very few people are working on drought, but salinity and fluoride, they're very
Shubho, your video is not working. Is it on purpose or?
it's so two questions joining at different time, so maybe the reason they
Shubho, the audio is not coming very nicely.
slide. Okay.
It's breaking, Maybe you could just
so there is
keep your mic
okay,
mic
I understand.
on and the video off.
No, I just turn off my camera.
But still, some, really struggling with the. It was going fine. What happened? Can you see if your internet is back on?
And
Can you check? Can you hear me? All right. So,
Shall I answer the question now or later?
No, we can start with the question answer round. And you can see that some questions are there in the chat, and we will paste if any more are coming in. You can stay. Yeah. So they are in the chat, in the Zoom chat. If you can see, you can read and you can answer them.
Okay. The first question is, as I can see, what is the role of DREP transcription factors and OSN family genes in salinity stress? I don't understand what is meant by OSN family genes, what you meant, but DREB, of course, is a very well-known factor. Now, DREB, you know that there are actually different kinds of DREBs. What is the full form of DREB? DREB stands for DRE binding factors or DRE binding transcription factor. I already told you, I talked about the ABRE or the coupling element sequences, but there are certain sequences which are also known as the dehydration responsive element, okay, which behaves in an ABA. Normally, they behave in an ABA independent fashion because there are certain genes, okay, which are induced by salt or which are induced by drought or even by cold, but they are not induced by ABA. So it's an ABA independent gene. So of course, the DREBs do act against drought stress or against cold stress. They have role in in the environmental stress tolerance. And what does OSN mean? It would have been better for me if you have spelled out the full form. OSN, sometimes it might refer to osmotin also. Okay, osmotin, if you really mean osmotin, then I would say that yes, definitely they are important because there is an osmotic stress in case of any environmental stress. There is a water shortage. So obviously these osmotin family genes, they are being activated during salinity stress because they have to ultimately restore the water level to dilute the the salt concentration. Okay, next question is by Shonali Bej. Sir, if we want to study the early response of salt stress, then what should be the time point standard? Should it be in hours or in days? Yeah, I think you should go in hours. Okay, because many of the early responsive genes, like for instance, polyamine biosynthetic genes, for instance, like SAMD carboxylase. Okay, it can be induced even in 6 hours. So it will be better if you go in hours, 6 hours, 12 hours, 24 hours, 48 hours. Okay, automatically you can, for early induction genes, normally it's better to go in hours rather than in days. Days will be quite a long time. Okay, next is Shangita Sharma. Sir, did you conduct any physiological analysis such as osmolyte accumulation in the transgenics? Of course, I showed you the data that when we overexpressed the RAP16A gene, both in tobacco and also in rice, okay, we measured different osmolytes. Osmolytes, I hope you know that the proline, even the polyamines, the sugars, reducing sugars, the total amino acids, okay, even sometimes for heavy metals, the cysteine, which is also an amino acid. These are very well known osmolytes. Yes, of course, you should do that. Okay. Next question is by I'm sorry, I cannot read the name properly. Soleiman, probably. He asked a question: by having a higher endogenous ABA level in plant while responding to environmental stress, can one be thinking that application of ABA may be a way of yes, of course. You can apply exogenous ABA. Even there are quite a number of work where you can apply ABA from outside. Either you can apply it in the hydroponic solution, where the plant will be able to uptake the ABA through the root system and accumulate, and thereafter you can check the different protective or the defensive mechanisms. Okay, but you should be very careful when you are applying this kind of exogenous mode or application. You should be very careful regarding the selection of concentration, because if your concentration surpasses the threshold, okay, the same protective agent which you are using can be very injurious. Okay, if you increase the ABA level to an extremely high level, then the ABA might act as a boomerang for you. Okay, it might not confer protection. So you should be careful. You should make a proper concentration dependent study. You should make a proper time dependent study, and then you go for the application. In fact, whatever work that I have showed, in the background, I have not shown those data, but in the background, it will need another 1 month to standardize all those concentration and standardize all those time. Okay, and then you come up with the conclusion. Ibrahim Kani Aminu, what is the effect of fluoride on rice growth? Okay, that's the entire scenario that I discussed. Okay, there is a lot of fluoride accumulation. Fluoride bioaccumulation is very high because the fluoride uptake occurs through the different fluoride channels, the CLCs, and then there is an overall damage. Okay, starting from the chlorophyll degradation, the growth inhibition. Okay, there is a lowering of the grain yield formation. Okay, then all other oxidative damages, like lipid peroxidation, membrane damage, electrolyte leakage. So overall, it does so many things, and overall oxidative damage. So the effect is very much disastrous on rice growth. But as I told you that there is a difference in the susceptibility. Not all rice varieties will be equally, subjective to equal stress. Some would be tolerant, some would be sensitive. This is the beauty of rice. Okay, this is the beauty of rice, that of working with rice, that you have so many varieties, you have so many germplasms, and you cannot come to a fixed conclusion. Okay, that rice is behaving like this or rice is behaving like that. No, you have to work with different genotypes to come to a conclusion. Okay, you cannot make a general conclusion in rice, and that is why it is so challenging and so interesting system to work with. Okay, next is I am Dr. Qasim. Question is that sub1 gene shows pleiotropic effect and also confers tolerance to drought. Does it also have any role in salinity stress? See, I'm not a very expert person on sub1 gene, rather because I don't work on submergence tolerance. I have heard the name of the gene, of course, sub1 gene. As I told you that, yes, whenever you are talking of submergence, okay, then there is some kind of, the anoxic condition. It's also a kind of like flooding condition. It creates some sort of anaerobic condition. So definitely it's a kind of stress. And as I told you that in abiotic stress, all the pathways are interlinked. Okay, all the pathways are interlinked, and one pathway will actually stimulate another pathway. So it remains to be seen, of course, that whether it will generate salt tolerance, but of course it will have some effect. Okay, because I'm sure that it will be activating some other defense pathways or genes, which will to some extent generate salt tolerance. But yes, of course, the level is important. How much salt tolerance is being generated, that would be important in this case to understand. Another question is Somnath Banerjee. Sir, your current research works are done or ongoing at Sensibility? Yes, of course, the works are going, and I already showed that the students are working, PhD students are working. Yes, of course.
It will be our last question.
Okay. Pritha Mukherjee. Sir, is there any possibility that we can introduce genes which act as enhancers so that the aroma and the salinity stress can be achieved in aromatic rice? Introduce genes which act as enhancers. See, the term enhancer actually, it has a different meaning in molecular biology, right? Enhancer means, probably you have used the term enhancer in a very loose manner. Enhancer actually means that when you use an enhancer, that means it acts both upstream and downstream. It's almost like the promoter, but there are certain differences with the promoter sequences, right? Where these are some sequences which you can add, which are bidirectional. You can use them in the upstream 5' region or in the 3' region. They are orientation independent. Okay, you can use them as direct repeats or inverted repeats. So that is what is actually meant by the enhancer. But if you mean enhancer to mean that you just want to enhance the aroma content, yes, the answer is definitely yes, because I have showed you the metabolic pathway. I have showed you the different metabolites which are induced or rather which are responsible for the aroma formation. So if you go for overexpressing those, those metabolites, or if you try to, as I showed you, if you downregulate bad H2, okay, because bad H2, if it is high, then it is going towards GABA, that is gamma-aminobutyric acid. So if you can lower down the GABA level, or if you can lower down the bad H2 enzyme, you can get higher aroma formation. So you have to check the metabolites which are responsible for aroma and use them for enhancing the aroma content. And it's a very challenging work, I tell you, because in fact, that could be one of the major areas in future. Okay, to increase the aroma content in aromatic rice varieties. Okay. I hope there is no more question.
No, that it's okay. It's fine.
Okay.
Now we are move on our interview section, but before that, I wanted to give you 2-minute break. It's a long talk and very interesting talk, and you've put so many energies in your lab work and for the presentation. It's wonderful, and many positive comments are receiving on your presentations and deliberation of webinar. So I give you 2-minute 2-minute breaks, and Shoma will make ready the interview questions. Within that 2 minutes, I want to share my screen and wanted to announcement or it's little bit that now this certificate link now should working. I will give you the password now. And this is our website where you get the certificate after 2 days from here on the top of the website. And there are a lot of talks are upcoming. You can browse and register what is your interest. And here is a question collection system. We are collecting few questions for our next talk for the Bansal sir, where you can ask your GMO related questions, because I see the questions and Transgenic is got the interested, and he one of the good people for answering your questions. And we are currently collecting the manuscript or article for our upcoming plant science journal. You can download here the instructions and can submit from 2 options. Here is a submission manuscript option, and another is a magazine pages. And these are the few questions you frequently asked by email. There are the questions and answer. You can go through, and it's automatically solve many questions. Currently, we are 7,000 plus subscriber in our YouTube channel. I request all of you please subscribe. This is our option for the speaker. And if you wanted to give your presentations or webinar, you can put your information. We will contact you. If your talk is suitable for Bioingene, we can go for that. So it's a short tour of our website, and we are currently we are collecting the issue for the January. There is a few instruction is there, so go for submission. It's free and open access international peer reviewed journal. So I go to the Shoma. Please do your interview session and on your camera. Okay.
Hello. It was a beautiful webinar. I want to thank the speaker personally for doing such great work with Govinda Bhog, because it is one of my favorite. Big fan.
Because in pulao and rice,
Can't live without Govinda Bhog rice. Okay. So, yeah. So, all right. So we will now start the interview session. Today is our 27th webinar of the series, and we are honored to have Dr. Aryadeep Roy Choudhury with us today. Welcome, sir, to the interview section of our webinar. Sir, you have done some amazing work in the field of rice, in the field of plant science research in general. So, we would love to know how you built your career. Could you please tell us about your journey so far as a plant scientist?
See, I have been born and brought up in Kolkata. So as Liberty already introduced my some of my early works, that after leaving school, I had a lot of option. But since I always loved life science or biological science, perhaps there is no other area in which I might have done better. So at that time, I chose life science or biological science after completion of HS. What should I? Where should I go? I should go for zoology or I should go for botany? Because at that time, when we were students, there were not such disciplines like microbiology, biotechnology. Okay, only they were just emerging at that time. Then biochemistry courses, these were not there. There were only botany, zoology, and physiology.
Hmm.
Now I will not do zoology, physiology because I have to go for animal dissection. Okay,
Okay.
and I don't like animal dissection. So
Mhm.
so the only option that
Fair
was
enough.
left at
Yeah.
that was left was botany, because that was clean enough. The dissection was clean enough, so I think that would be better for me. So with that, okay, I went for botany.
Hmm.
Okay, and I was fortunate to get admitted to the probably the best college during that time, that is Presidency College, of course, where we got some of the good professors, okay, who actually built our shape or actually built our future. Okay, some of the great professors and teachers, and they motivated me particularly, and so somehow I finished the BSc honors in botany.
Mhm.
And then again, there was again a very wider avenue for me, because fortunately, since by God's grace, my result was quite good. So I had the option whether I will go for botany or I will go for some interdisciplinary subject.
Hmm.
Okay, like molecular biology or biophysics, which is actually taught at Rajabajar Science College, Calcutta University. So
Okay.
I had that option. So again, the parameter for measuring my subject was that, because a lot of people actually qualify for net in that department. So I
Hmm.
thought that, okay, chalo, that this could be my I think that could be a better destination, because at least if I get an MSc degree from this department, maybe there is a chance that I can qualify net. Okay, otherwise there was no special intention of choosing molecular biology. Of course, I like the subject genetics and molecular biology, but that was one of the main parameters to choose molecular biology.
Mhm.
Okay, so that way I finished the 2-year course and then qualified net. And then again there was a choice for me, lot of choice, because when you qualify MSc in molecular biology, you see that you can work with cancer biology, you can work with bioinformatics or structural biology.
Mhm.
Okay, you have so many options. You can work with immunology. Okay, now there was my main this for the first time. I asked myself, or I asked to my heart, that which is the area that actually is my cup of tea.
Okay.
Okay, because at that time, if I would have chosen immunology, I would have chosen cancer, because now when I teach, when I am a teacher in my college, okay, I will just come maybe later on, maybe on that issue. the
Mhm.
I find very few students they are focused in plant science. Okay, so
Mhm.
that was at that time I chose that, okay, fine, this is my cup of tea. I will work with plant science, because uh, our country is basically an agricultural country, and it needs input from a large section of scientists, okay, to improve the productivity, to improve the agriculture. Okay, so
Okay.
that was my decision, and I continued with working with rice, which is the main staple food crop. The credit goes to my supervisor. And then during my postdoc, I worked with, you can say, the leading rice scientist of the world, you can say, Professor Shapundottu. And yes, so I was fortunate to join his lab and work with him, along with, of course, Madam Professor Korobidotto. And finally, again, by God's grace, somehow I got the chance to join as assistant professor at St. Xavier's. So that's how I'm continuing there, continuing with my research, and trying to follow the footsteps of my teachers, my supervisors, PhD and postdoc supervisors. Okay, trying to follow their footsteps. That's it.
Oh, wonderful. Do you think we should consider abiotic stress more seriously to ensure a global food security during the times of climate change and global warming?
Definitely, because the climate change has become a severe problem in the present times, and we already know of the global warming and the greenhouse gas emissions. Okay, the ever increasing rise in the carbon dioxide level in the atmosphere. So these are some of the issues which are very common, but and of course we need, and that's why those kind of projects are very important, where you try to increase the carbon concentrating mechanism even in the C3 plants. So you try to find out what are those genes which are present in the C4 plant, and try to overexpress those C4 genes in C3 plants, so that they are photosynthetically more efficient and they have a better carbon concentrating mechanism. But I tell you another issue which is also becoming very severe in the present time, and that is we should also try to draw our attention or shift our attention to another increasing problem, which is also a greenhouse gas, and that is methane.
Oh, yes. Mm-hmm.
Because if you go through certain reports, I mean media reports or newspaper reports, you will see that in the Antarctic region, there is in the recent times there has been a lot of methane seepage. Okay, because of the global warming, because of the melting of the glaciers. Okay, even in certain portions, the coldest regions of the world, like Siberia and Alaska, they are facing a massive global warming. It is because mostly due to methane emission.
Right.
So I will not be surprised if within another 10 years we will be hearing something like methane resistant plants. Okay, because probably we will be needing some overexpression of genes from maybe methanogens or maybe methanogenic bacteria, and we need to fish out those genes and clone them in the crop plants. So that time is also not too far. So of course, developing climate resilient plants. And in fact, I have framed another book, and that book is also being approved by Elsevier, and that will be on climate resilient plants. So that is an approved book. The contract has been signed, and there we will try to actually, yes, we will be, thank you, we will be trying to address these issues. Yes, so it's definitely very relevant and very contemporary type of, you can say, work. Okay.
Right. How do you encourage your students towards plant science research?
This probably is a very, very difficult question. Why difficult question? Because see, I'm a teacher, and I see the students from or what should I say, from a very close approach. I see the students, the MSc students, because ours is the postgraduate department, and St. Xavier's College, we all know that it's the leading academic institution in India as per NIRF ranking and all. And we have very good students already. We have very bright and motivated students. But the problem with the students is that only 1% of them, okay, they are interested in plant science. Whenever you ask students, okay, that, well, what would be your research area? Obvious answers which come are cancer biology, immunology, developmental biology, neuroscience, okay, few structural biology,
Yeah,
and
trendy.
there are some vague answers. Yes, and there are some vague answers. I tell you, because this is a platform that the students are hearing, and I want to really address this issue. Most of the students also answered like, Sir, I will do molecular biology. Sir, I will do epigenetics. Sir, I will do biotechnology. Now, these answers are very, very vague. What is meant by molecular biology is a tool. Biotechnology is a tool.
Yes.
Epigenetics is a tool. A tool or a technology cannot be science. You
Right.
have to
Right.
know the system. You have to know either microbial system, you have to know a plant system or an animal system. Your technology cannot be science. So the students are confused between a science, a concept, okay, and the technology.
So true.
So the students who are listening on this platform, please make sure that you know the subject. Okay, in biology, if you look at the subjects, you have three basic subjects. One is microbe world, one is plant world, and another is the animal world or human world. So make sure which of these three worlds or four worlds attracts you, and then you have to choose a system. Okay, and it's really hard. The question is very interesting because it is really hard to motivate the students towards plant science, although we know that the agriculture is the mainstay in India, even maybe in abroad, the food production, because you know that in this pandemic era, if you sit at home and if you have no food,
Yes.
okay, so you need to go for production, you need to improve the agriculture, and the entire country or the world need bright students like you to join this wonderful world of plant science and agricultural science. Okay, so I try to motivate the students in the best effort, but ultimately it's their choice. Okay.
Right. Um, what motivates you to work for the betterment of agriculture?
Lot of things, because as I said, we have so many problems in agriculture, like nutrition. Malnutrition is one of the major problems. Okay, so going for the biofortification programs, enriching the different plants with the different nutrients, study the nutrient profiling, make a nutrient profiling of the different crops, or antioxidant profiling of different crops. So that is one of the challenge. Of course, any kind of stress, not only abiotic but also biotic stress. So we need to have a sustainable agriculture. And one more thing is that, till date, what is the best technology that we have in improving the crop systems? The answer is transgenic, right? Because there are several criticisms against the transgenic generation of the transgenic. But I have only one humble question to those who are criticizing this technology: that if you criticize this technology, then you have to come up with an alternative strategy.
Yes.
I can criticize any technology, but at the same time, I have to give the solution.
Right.
Otherwise, there is no point in criticizing. Now, when you ask them, then what is the solution? They have no answer. So if you have no answer, then it's better that you keep quiet and let the scientists work and let the scientists continue with the technology. So that probably is one of the, and another thing is important, that the plant physiologists, the breeders, the geneticists, the molecular biologists, the biotechnologists, all of them should work together. There is no, I mean, there is no, that breeding is a different issue, molecular biology is a different issue. Okay, even the people who are working in the seed companies, okay, they should work in unison, and one should not consider each other as a rival. Rather, they should work together and make a good team and work. So that should be the motivation, and in fact, that is
In
also my motivation.
collaboration.
Yes, collaboration, and that is also my motivation towards working with plants or agriculture. Yes.
Okay, so talking about collaboration, you have edited so many books which require strong collaboration with many authors. How do you manage it?
It's the answer would be the same as how Shubho manages so much wonderful network in spite of his difficulty in spite of this busy schedule and working in the lab, okay, and fulfilling the demands of Professor Dotto, okay, it's almost the same answer. It's almost the same answer. Okay. Actually, I do it for my own benefit, you know, because when you do this kind of things, okay, you get to study. You have to study also. Okay, when you write a proposal, book proposal, you have to make a proposal and you have to submit. You have to study. Moreover, you get writings from so many authors, which means that you have to read those articles.
Oh, yes.
Now, when you read those articles, automatically I am also enriched. So indirectly, they also help me in, you know, enriching my knowledge. Okay, because though I am working with salinity and fluoride, that does not mean that that is the only area. I should also know about the other areas. Okay, so actually, I do it for my own benefit, you can say.
Right. You are a reviewer for so many renowned journals. Can you give us some key takeaway messages to improve manuscripts?
Okay. One thing is that your manuscript, it should have a clear cut hypothesis. When you are writing a manuscript, especially this I'm telling for the beginners, it should have a clear cut hypothesis. There should be a clear cut aim, and it's very important that you follow the author's instruction. What is the scope of the journal? Okay, because sometimes you know, without going through the scope, you submit the manuscript. Okay, and you are wasting your time, because you are wasting maybe 1 month time, 2 month time, but it's not within the purview of the journal. So that is very important. The hypothesis, the quality of the figures, that should be very important. Okay, clarity of the figures. And another very important, which most of the manuscripts nowadays ask, is very proper statistical treatment.
Right.
Because nowadays you cannot publish good papers until you have those kind of you made the, you know, the sampling analysis, or the ANOVA or any kind of statistical treatment. That is very important. That has become mandatory, in fact.
Right.
Okay, the number of replica that you use, okay, and the statistical sampling, all these treatments, those are very important. So you should be careful. And another thing is that the language, the writing skill, that should be improved, because, you know, in science, it is very important that you are not only a good bench worker, but you should also be capable of writing the manuscript, because you need to represent it properly. That is also very important.
Right.
So these things should be kept in mind. Yes.
Yes, it's very important to communicate your work properly. The work which you
Yes.
did, so so much hard work, needs to be communicated to the world.
Communicate. Yes, definitely. Yes.
Okay. So according to you, which sectors of plant science research will prosper in the near future?
Number one is climate. As I said, that climate change is already there. So climate resilient plant, developing climate resilient plant. This is number one. Number two is that I was hearing, in fact, in your Bioingene platform, I was hearing about one of the works. I think that was one of the work by Dr. Anindo Bandyopadhyay, who was focusing on that biofuel.
Right.
I think, yeah, probably. So I think that is also very important area, because if you really want to cut down the greenhouse gas emission, then you have to stop reducing fossil fuels, and you have to go for or you have to choose those kind of plants, or those kind of microorganisms, which are a rich source of, the diesel or biofuel, which can produce this kind of. So I think that kind of research, okay, exploiting plants for biodiesel production or biofuel production, is one very important area which is which is important. And CRISPR-Cas technology, this is also, I think, is an upcoming, yes, because this is gaining more and more popularity nowadays. Okay, so that technology, I think, will also gain priority.
Right. All right. So we are nearing the end, and I have one last question for you. What would your words of advice be to the students and scholars who are new to the field of research?
Number one is research is not a time pass. This is my first message. Okay, that I am not getting anywhere. That is why I want to just spend my time in research. This should be rather, it should just be the opposite. I am getting all other options, but still I have come to research.
Yeah.
This should be your main target. Okay, and one should be asking question that whether research is really my cup of tea, whether I can really spend 5 years, 10 years, long time in research, PhD, and then postdoc, and then maybe getting a job. So of course you should have a family backup also for that. Okay, so that should be taken into consideration. It's a long time. It needs perseverance, patience. Okay, because PhD training is not just learning few techniques. It teaches you so many things. Okay, it teaches you how to work in a group. It teaches you how to be patient. It teaches you how to behave with your seniors and with your professors. Okay, so it's an overall ethical value and moral values which you learn from your PhD curriculum. Working in a group. Okay, be calm and patient. Okay, try to be loyal. So these are some of the things which are very important to learn. So whether I am really fit for this system, that question needs to be asked, number one. And number two is, what is my subject? When you are in the first year, you say that my subject is immunology. When you are in the second year, you say, no, 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 I am in structural biology. In the third year, you say, no, no, developmental biology. Then in fourth year, plant science. And when you finally finish your PhD, you again go back to cancer biology. So if you have such a fluctuating mind, and when you join a research, you say, no, 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 research is not my cup of tea. I would go for IAS training or IFS training.
Mm.
So if your mind fluctuates in such a fashion, then you will be nowhere. So it's better not to come to research. You should have a very fixed mind with a fixed goal. Okay,
Yeah,
and
research is not for the faint-hearted.
yes, yes, you should be really, really committed and serious. Okay,
Yeah.
for and then only come to it. Otherwise, you are free to join any other, any other job or any other
All right.
courses.
Thank you so much. It was a wonderful session, and such beautiful work, and such well explained and well illustrated. Really, really, really honored to have you, and so many appreciations. We are getting on our YouTube chat. Thank you so much. Shubho, would you like to say something to our speaker? I don't know if your mic is on mute. Can't hear you. Maybe you could just keep your audio on. Can't really hear you. Still can't hear you.
Can you hear me now?
Yes, now I
Yeah.
can hear you.
So yes, sorry for the internet glitch. I don't know why it's happened. It's maybe 31st, and so
Last day, the internet is saying, "Give me a break now."
okay. I'm very honored and happy that today a lot of questions is arise for the point of view of students, for the point of view those are asking more questions and more doubt about GM technology, and the field like the speakers and our sir says that it's a vague. Yes, and nowadays I'm looking in some craze about the CRISPR, but it's
Yeah.
not the why they wanted to be CRISPR,
Yeah,
but
we had
just
questions
I
also,
wanted
na,
to
like that.
CRISPR.
Yeah. Like they want to do
So
PhD in
my
CRISPR. We
point,
had questions like that.
Yeah.
yes. So my point of view is quite same, and this is the reason that we were trying to build Bioingene to collaborate and interact with others plant scientists and other students. That plant is a very beautiful system. So we obviously we are working on plant. That's why it seems to be a partiality about the plant, but it's lot of thing you can do, and more serious things that what we are focusing, as you see in our website also. We are focusing, we very challenging is the food security, nutrition security, and fiber security, obviously plant fibers. And I'm also working on fiber crops. That's why I little bit know about its importance in economic and commercial sections. So my point of
Shubha, getting an echo.
excuse me, yes,
Yeah.
I try to stop. Is it okay?
Yep. Good.
Okay. So it's very important field. I request many for those are the BSc and MSc students. If you consider a research field, then you may consider plant as a field, and obviously any research field need very passions and very dedications on that, and where same for the plant, because it has a generation time. It's not a bacteria for overnight growth.
Yeah.
It has a specific generation time, and when you working an annual plant is very difficult. Like I am working on a jute, and it takes seed to seed for 10 months to 1 year, and that's why I can manage other works too. When my plants are growing in greenhouse, I can manage others work, writing, working on biofortification of rice, or during the lockdown, it's Bioingene. So it's a collaborative work. It's not me, my friend Shoma, Jacinta, Liberty, Neha. They all are, we are trying to collaborate in such a way which benefits the people. And in my MSc, BSc, I'm not getting such platform where I can interact directly to the speaker. Even I can ask whether you are doing your research or over in your lab. Like today, I took a questions that this is this type of questions, and I got many others related questions. I wanted to ask one last question from our speaker. It will be a concluding remark. Like one student, obviously, yeah, I think he's a student. He's Somnath Banerjee. He said if someone does MSc from a college instead of a big university and qualifies CSI net later on, will the student face any problem during PhD? So it's a combinations of your knowledge, your intention, and your motivation. So I ask our speaker to answer these questions. Is it college is important, or motivation is more important in research?
The very simple answer is your institute does not matter at all. Okay, if the institute matters, or if the place where you study matters, okay, then we would not have got some people like there some of the great people who used to, maybe their childhood days they used to live in remote villages. Okay, and then also if you see in the long run they are quite established. Okay, there are many such examples. So it does not matter at all where what is your family background, or which school have you studied, whether you are English medium or Bengali medium or any other Hindi medium or regional medium. Okay, or whether your college is a renowned college or your university is renowned. The only thing that matters is how much effort you have put, okay, and how much dedication you have. That is the most important thing. So if even if you are studying in not that much well-known college or university, just don't be scared of that, or don't feel shy. Okay, you can do good from any institute or from any university or college. Okay, just focus on your work, focus on your studies. Okay, and try to make the best benefit and try to do good. That is important. Okay, so don't bother about all these other, which are, secondary issues. These are very secondary. Yes.
Okay. So thanks once again. It was a nice, and you covered so many aspects. Not only your talk, not only the science. It's covered so many other aspects which we also try to cover in the bioengine. Thanks for coming. Thanks for delivering your talk. We hope in near future, when your some work already in the published, and we love to hearing you again in Bioengine platform.
Sure, sure,
Thanks for
sure.
the joining for today's webinar. I wanted to thanks every participants, those are watching this live session. Uh, Thanks for joining Bioengine, and I wish a very very uh, uh, year ahead. Happy New Year in advance. Is the last day of this year,
Yes.
and
Yes,
Hope,
hopefully next
yeah.
year brings
Hope this is a
so many
better
positive.
year coming.
yes, yes.
Yes.
So this is
Great.
really,
Thanks,
this is
Soma
also very,
Liberty, for helping me to conduct this webinar. And there is some internet issue from my end, or somehow. So I apologize for it, and I'm sorry. It's not my hand actually. So thanks once again, Shoma. I request you to end this session and you can you share the screen and ask people to submit our bioengine PSJ in our journal and upcoming webinar. And there is a option, those are interested in GMO and have a GMO related questions, so we are
Yeah,
collecting the
sure.
questions. And thanks,
Yes.
Ajuda, for
Arjuna was saying something. Were you saying something?
yeah. Before I leave, this is also a very auspicious day for me, because I am the last speaker for this year. Basically, I am ending this year,
Yeah.
okay, on the Bioingene platform. So it's also very remarkable and very auspicious for me. Anyway, I wish all of you, all the viewers, okay, all the students, the entire unit of Bioengine, starting from Shubho, Shoma, the Liberty, and all others who are there in the Bioengine, a very, very happy new year ahead. Okay, and let's pray together that the coming year, at least we get rid, as early as possible, we get rid of this situation, get rid of this pandemic situation, so that we can return back to our normal
Yeah.
life as
Yes.
early as possible. Okay, so okay, I wish all the success for this platform. I wish all success for Bioengine. Okay, okay.
thank you.
So
Thank you.
shall I leave? Okay,
Yes,
okay,
yes, yes,
Sure.
okay,
yes. Thank you.
okay,
Thank you,
okay,
sir.
Uh,
okay.
yeah.
Thank you.
Thank you. Welcome.
Thanks,
Bye.
Liberty.
Thank you, Liberty.
No
Thank you.
problem.
Yeah. It was lovely to see you on the, it's already 31st here. So it was lovely to see you on the last day of the year, and hope
Yeah,
we have.
you're in the future.
Yeah, I really hope we have a wonderful year ahead, and have, like, Bioengine grows and more people join us now. And also all the best for your career, good things happening in your career as well. Yeah, you're almost
Yeah,
completing. Yeah.
I
You'll be
don't
a
know, but
Dr. Liberty Galvin soon.
yes. That'll be very nice. Thank you,
Yeah.
Shoma.
Right.
Hoping
So
to see you in 2021.
Yes. All right. So
Yes,
I'll just
same to
do
you.
a little bit of the new website and the call for papers,
Have
and then
fun.
I'll.
Okay. Sounds great.
Yeah, sure. If you want, you can maybe take rest.
Rest.
Okay, bye.
What's rest? Okay.
All right.
Thank you. See you soon.
Yeah.
See you soon.
Bye bye.
Bye.
All right. So your as per your request for the alpha papers thing, I'm going into bioingene.com. Okay, I'll just share my screen. All right. So how is it looking?
Yes.
Okay, so I just shared my screen, and so all of you can see that our website is here, and we are promoting our first webinar of 2021, that is on 11th January. And you can see there's a screen flashing, that is ask Dr. Bansal, Dr. Casey Bansal. He is the person who would give you authentic and answers for your GMO related questions. And because GMO and GM food related questions are there in every person's mind, that is it good, and is it because sometimes we see that it is being advertised that these are non-GM, and as if it's a good thing, like is as if GM is a bad thing, so it's being said that in packets and in food packets it's non-GM. So is it such a bad thing, and then why is so much research is being done? So you get your answers on 11th January. All right, so register for that event. It's going to be an amazing event, and so much to learn, so much knowledge, and inspiration and perspective. So moving on to our magazine. So we have been promoting it for some time now. We have launched our initiative, that is the Bioingene Plant Science Journal. You can click on the magazine section on our website, and you can see that the description has been given here about the journal, a little bit about the journal, and what are the topics that are going to be addressed here, what are things we're going to publish. So it's an online, international, open access, peer reviewed scientific journal. It's a monthly journal, and it's completely free. That is the thing, it's free for everyone. No, you don't pay anything, you just have to put in good research, well written and good research. You just give that, and it's going to be published, and has to be original, and all the criteria that a journal holds, that you have to see before you put in your manuscript. So you can also get the guidelines here, and please give it a reading. All right, so there's a submit manuscript section. If you scroll down, you will see there's a submit your manuscript. You can submit it here. Like, I clicked on it, and there's a Google form opening, and it's all your details. You can put in your details here, the title of the manuscript, your email address, a type of writing, type of article or write-up that you're submitting, that is a review article or summary of a published article, concepts or policy forum, if you want to add your ideas or your thoughts about a policy in agriculture or plant sciences. Then you write in your title of the manuscript, and what are the keywords, and your abstract. You paste in your abstract here, and this section, the DOI number, this only for summary of a published article. So if you don't have any original research that you can publish right now, you can doesn't matter. We are also taking summaries. Like if you read a current um, article on plant science from a reputed journal and you really loved it, you liked it so much that you want to write it in your own words, write about it in your own words, you write about it, write it in a way that everyone can understand, not only people in science but everyone. Don't put in technical jargons, write it in a very simple way so that people on all of the world can understand it. And that is the way science can reach all the people. If you take out the technical jargons, all the difficult words, scientific words from it, and just put it in a way you can understand, a simple person, a layman would understand, then that you're doing a favor to that to science. You're really doing a favor to science, that you spreading the current research to the world. So you write a summary and put it here. Okay, so then the next comes the author's name of the corresponding author, contact info of corresponding author, upload your write-up or manuscript. You can upload, add a file here, add the you can add the PDF or Word file, and you can include text, table, and figures. Also, if you need guidance regarding how your figures should be, you can watch the video which Shubho had presented. How to prepare your figures for your manuscript. You can watch the video. You can get some help there if you require some help, like how should my figures be and how should I design them. So here's an email address again for your correspondence, like the institutional email, preferably from the institute, authentic institute email. And then retype that email again, then your name, then your country, and then you read out these declarations, and then you confirm, then you submit. It's simple. All right, so going back to the home page. So you can see this flashing, like the screen, it's a dynamic screen is coming and going. So it's Ask Professor Bansal. So you can just click on it, and you can ask your questions. Like I'm repeating this. If you have questions regarding GM crops, GM food, you can please type in your questions so that we have all the questions and we are ready, and we can ask our speaker. And also you can send the questions to our speaker so he is prepared and your questions are being answered that day. All right, so is there anything else that I needed to say?
No, it's okay.
Yeah, if you have any questions regarding more questions regarding your the Bioingene Plant Science Journal, you can write to us. Definitely, we will try to answer your questions. And yes, it is free. And if you have original research, you can publish. That's the thing. Even MSc students can do that. Okay. All right. So, how about ending the webinar, the event? If you want to say, do you have anything to say? Yes, it is free. And if you have original research, you can publish. That's the thing. So, even shall I?
Yes.
Your voice is breaking.
Thanks. You just end the session.
Yeah, sure. Okay, so yeah, it was a good session, and see you all in the next year on 11th January. Yes, please register.
Okay. Thanks everyone. Thanks everyone for joining today's webinar. Wish you happy New Year. We are again met in 11th of January. Thank you.